between eukaryotes and prokaryotes, let's look at the packaging of DNA again. And we went through this earlier. As you would see here, this is the folding of the DNA and packaging, unfolding and packaging of the DNA from this level of a chromosome. And here we talked about the nucleosome here, which is made of 8 histones. And this is the H1 histone. And then we have the chromatosome, okay, which is the nucleosome, this part plus the H1 histone together. So this folding and brings together, if we want to try to visualize chromosomes that we have, okay? And next, as we try to explain differences between eukaryotes and prokaryotes, and talk about for cell division, for example, which splits a chromosome into chromatides. Okay, this is one chromatide, but it's not sister chromatide. What you can see here is, perhaps you can tell from the right side that this is it's a photograph. It is kind of image of the chromosomes from a single cell. Okay, and it's usually arranged from largest to the smallest. Okay, from largest to the smallest, and we refer to this as a karyotype, and it is basically a graph, a visual representation of individual chromosome. And this is another way to visualize that chromosome, which is called ideogram. It's just a schematic representation or diagrammatic representation that illustrate the different characteristics and features of chromosomes. And sometimes you can have access to online visualizations or interactive visualizations for a chromosome, where you can hover and get more details about what's in these chromosomes in terms of genes and so on and so forth. So eukaryotes and prokaryotes. Eukaryotes and prokaryotes. Prokaryotes are those one or single cell species, right? And we look into this. We have went through DNA replication, and it is a kind of involved process. Before we look into cell division in eukaryotes here, so I want to take you quickly, because in prokaryotes there is what's called binary fission, okay, in eukaryotes, which is a method for asexual reproduction for the prokaryote organisms. I'm talking here referring to bacteria, for example, to archaea, and so on and so forth. And in this species, the single cell divides into 2 genetically identical cells. However, before it begins divides, the single cellular DNA molecule is replicated. So the DNA molecule is replicated before the cell divides. And then there is what's called cell elongation. This is what you find here, the cell elongation, and keeps going until it makes of what is called formation of sperm, okay? Formation of sperm. And at this stage, here we have an involvement from the FTSZ proteins, trying to help with moving away the cells, like pushing them towards 2 different poles. And then we have the formation of a sperm or a partition, okay, or a partition. You can see here there's some kind of partition start forming as compared to this ring, which is called FTCZ ring. And approximately the sperm is at the midpoint of the elongated cells. Eventually, these 2 cells are separated. Okay, so this is summarized the cell division in prokaryotes, the single cell species. Now, moving to cell division in eukaryotes is actually complex. is a more complex process, okay? And this involves different phases and stages. So traditionally, there are 4 main phases that are called G1 phase or gap 1, S phase for the synthesis, G2 phase, M phase for mitosis, and so on and so forth. So this will take all the stages for the cell growth, preparation for DNA replication. Okay, we have and after that. DNA replication actually takes place in the S phase or the synthesis, and then we come to the G2 phase, which follows DNA replications, and which follows the DNA replications and precedes the onset of mitosis. And then the cell prepares for mitosis by synthesizing proteins and other molecules needed for the segregation of the cellular contents, and that is what's referred to M phase. And the mitosis is further subdivided into prophase, metaphase, anaphase, and telophase. Okay, so in general, the cycle is of the cell division in eukaryotes is regulated by a series of checkpoints that monitor the integrity of the DNA. That is G1, G2, and M phase checkpoint, G1, G2, and M phase checkpoints. Okay, and helps in understanding or in knowing the integrity and making sure that this is a proper progression of such events like DNA replication. So further, and in addition to this, when we come, we had looked into cell division. Now, what about DNA replication? And for DNA replication, prokaryotes are circular chromosomes. So you remember when I've been showing you an example, just, for simplicity, I said that kind of virus kind of circular chromosome, and talking about one origin of replication. In fact, this is one thing that characterizes prokaryotes as compared to eukaryotes. Eukaryotes are multiple linear chromosomes, and there are multiple origins of replications, initiation sites. And this table here gives you a list of interesting ideas that differentiate between prokaryotes and eukaryotes. For DNA synthesis, there are different types of polymerases for primer removal, for RNA to DNA replacement, and telomerase synthesis that we have discussed earlier. Now, one thing I want to highlight here that is the DNA polymerase delta, which is one of the essential DNA polymerase in eukaryotic cells. It plays a major role in DNA, as you would see here. It's DNA replication. Okay, DNA replication. Okay, its essential role is specific. If you review back the discussion on the lagging strand and Okazaki fragments, okay, that is where this polymerase comes into play, specifically in not just only DNA replication, it actually participates in the DNA repair process. And this highlights another set of difference between eukaryotes and prokaryotes. Another thing to look into is to dive deeper into or zoom in into areas of the gene, and here we can see some interesting things. So there are several genes here with a terminating end, and they all share a promoter region. Now, this is unique to prokaryotes, okay? And this set here of genes are referred to as operon, okay? Which is a functional unit of the genomic DNA. It is referring to a cluster of genes under the control of single promoter. Okay, there is one promoter controlling these this group of cluster, or this cluster of genes, or this group of genes. Okay, we're going to highlight one example for a traditional example for operands, which is called lac operand. So we refer to this operand as the gene structure or gene area for prokaryotes. Now eukaryotes which represent, for example, the way that we have our genes structured, it has a continuous stretch of codons in this area. There are a group of introns, exons. There's a poly-A signal sequence here, and there's a promoter here. This might code for one specific gene eventually. Okay, so it may has the potential to be translated, in other words, to a specific protein, to one specific protein. Okay. Now, lac operand it's part of the E. coli bacteria. As I said, is one of the traditional classic examples to demonstrate prokaryotic gene regulation. So just going to give a quick summary of this here. The lac operands control the breakdown of, so again, this is an area of gene, and that is part of a prokaryotic, okay? And that is now gonna be exposed to some energy sources, so lactose, okay? Lactose. So the lac operands control the breakdown of lactose. In other words, sugar, okay? We know that lactose is sugar. So it's a source of energy. And so when E. coli as a bacteria is in an environment with lactose, it needs to produce some enzymes to metabolize it, okay? So there are 3 genes responsible for this, and the operand LAC-Z, and which is going to be responsible to convert lactose into glucose and galactose. Okay, there's LAC-Y gene, okay, which helps in transporting lactose, and there's LAC-A gene. Okay, these are 3 genes that they share the promoter area, and the expression of lac operand, this set of genes, okay, this set of genes is regulated by 2 main proteins. There's the lac repressor protein, okay, lac repressor protein, and catabolite activator protein. Catabolite activator protein. These are 2 areas here. Actually, the proteins are here in the slide, and here is the repressor protein. Okay, and giving this setup, because there's the catabolite activator protein, or CAP for short, which plays a role in the cell's preference for glucose over lactose as an energy source. So this is going to give us scenarios. So if glucose levels are low, the what is called cyclic catabolite activator protein levels rises in the cell. So what happens in that case? Okay, so we can see here in the slide there are specific scenarios. Like, for example, what if we have low glucose and lactose is available, is present? Okay, and in that case, the lac operand, this area, okay, is transcribed at high levels. It's transcribed at high levels. As you see, there are a set of actions happening here. RNA polymerase is going into action, and so on and so forth. So it's transcribed at high level, and the absence of glucose means high CAMP levels, allowing CAP to activate transcription. So both this protein here, the RNA polymerase, is going to play a role and give lac strong expression. It's going to be strongly expressed. And then we have the other scenarios, which is going to be, in this case, we have high glucose or low glucose, but lactose is unavailable, and in both of these cases, LAC genes are not expressed. And then the final case of high glucose and lactose available. So these are three genes responsible for generating the messenger RNA. And again, depending on other set of proteins that regulates the expression here, we can see this example how E. coli is going to benefit from the surrounding energy sources. If we move ahead with eukaryotes versus prokaryotes, one things that makes eukaryotes unique is that eukaryote genes can have proximal and distal regulators on transcription. So here's the gene A. How and upstream side we have the promoter where RNA polymerase come into play. But see the enhancer, these are control elements that would influence the gene, but they are not close to it, okay? They are far to it, right? Far away within the coding region of the transcript, within the coding region of the transcript. In particular, the concept or the keyword of enhancer in this case, enhancer, it is a DNA sequence that is going to promote the transcription of a specific gene, and it's usually made of short DNA sequences. Okay, that are these distal control elements. Now, finally, here we have this next. Yes, so and yep. Sorry, so it's not final yet, so we're going to come to that. A couple of more slides, a little bit more patience, but still we're highlighting, and summarizing a few of the main major differences between prokaryotes and eukaryotes. What I want to highlight here in terms of transcription initiation, transcription initiation, the area or the site responsible to unlock the expression of the gene and start the activity there and the translation process, it is different between prokaryotes and eukaryotes, okay? Prokaryotes, they have specific promoter genes. These promoter genes, like they are 10 and this up, like 10 genes upstreams, TTAAT, okay, and the 35 minus 35 sequences. So we're going like minus 35 steps, part of the promoter regions, and so these areas makes the promoter makes up the promoter in prokaryotes, and that is a unique composition and shape for them. And another thing that we're looking at as well is this other area in eukaryotes, okay, what is called Tata box, okay, Tata box. So it is a more complex structure for the promoter in eukaryotes, as you would see here, as compared to these regions, like in the upstream area, minus 30 genes we're looking here, we find the Tata box, okay? Eukaryotes use 3 different RNA polymerase, each for different gene types. So there is RNA polymerase 2, responsible for protein coding genes and requires multiple transcriptional factors, something we looked into earlier, right, for initiation. We were looking into transcription. And also eukaryotic chromatin structure must be modified for accessibility. And these adds more to how this area is shaped in the promoter of eukaryotes. Now, eukaryotic transcription does not have a specific termination sites, and this is a very interesting concept And I guess a way to highlight it, you know, somebody is running here, okay, trying, but there is no end. There is no end. And this guy is, you know, have energy to keep going, and there's no end. Now, what would you do if you want to stop this guy? Think about it. Okay, what would you to stop this guy? Okay, you're gonna come here, and you need to be much faster than this guy, or faster than this guy. And once you reach out to him, you push him to get out, and this is how you would stop him. Stop and say stop. Okay, in that case, I would put the colors back again and say we're gonna send this guy back, maybe back home. Okay, back home. So similarly is this, what you see in this figure, that's other guys actually is what makes what's called the torpedo model, okay, which is the mechanism of how transcription terminates in eukaryotes, which is different than prokaryotes. Unlike eukaryotes transcription does not have a specific ending or termination, okay? So what you see here as the RNA polymerase is working, okay, as RNA polymerase 2 transcribes the DNA, here it's working, right? It eventually encounters a sequence that is going to guide it to add the poly A tail, okay? The poly A tail, here's it, okay? The poly A tail, okay? It's going to encounter that sequence. They're going to tell us to add the poly A tail, which is, as we said earlier, a string of adenine nucleotides to the RNA. This side is cleaved then, and the poly A is added to the nascent RNA transcript, okay? Now, after this cleavage, RNA polymerase continues transcribing, okay? So it keeps going. Remember the guy who keeps, you know, keeps working, okay? DNA polymerase never stop. So what is going to happen here? Something is going to happen. There's what's called torpedo RNAs right from here, right? Think about it as some object here, okay? It is actually an exonuclease enzyme. It's an enzyme, okay? That is going to come and degrades the unnecessary downstream RNA. And so keeps eating this unnecessary RNA and keeps eating it until whoops, it's gonna push it to stop, as we had in our previous example of both guys that they were running. And so now it stops, and RNA polymers know that it should stop now. And so poly A tail was a signal for that torpedo to jump, and then the termination to happen. Okay. Another interesting thing that I want to highlight here in eukaryotes, you can see in eukaryotes, they have exons, introns, and when there's transcription, introns goes away. However, the arrangements of which exon come after which, so in this case, this may come first, this may come second, this third, or another combination could happen. This come second, this come first, and this third. So these different combinations are what define what is called alternative splicing, okay? Alternative splicing. And so one of the most significant contributors to transcript variability. That is why post-transcriptional modifications allows for transcript variability. The fact that exons can be arranged differently, for example. 5' capping, it is the 5' end of the pre-mRNA, which receives a modified guanine nucleotide cap. And this is certainly essential for stability. However, certain modifications can also influence transcript variability, okay, which eventually affects also the stability, and so on and so forth. So given the differences between DNA and RNA, as you understand now, what could be a potential target for selectively capturing mRNA? think about all of these concepts that we understand. Take a moment now and think deeply about DNA and RNA. Let's think deeply about that and try to answer this question. How the process goes in terms of transcription, okay? And as we get messenger RNAs, we want to capture them. And one hint maybe why you want to capture them is when I measure gene expression, okay? So what could be a potential target to capture? So I want to give you here a moment, okay, to think about it. And again, note this question down and share with me your answer for this. So one of the most distinctive features of mRNA that sets apart from DNA is the presence of poly A tail, okay, which comes really at the it is this stretch of adenine nucleotides added to the 3' end of the precursor messenger RNA, okay. And finally here, what I highlight is protein synthesis is similar across organisms. That is the translation set. So you can see a very similar process in between both of these figures. Yet the ribosome subunits, the ribosomes that take responsibility for the process of protein synthesis, are different in terms of measurement, okay? So prokaryotes are dealing with 30S and 50S ribosomal subunits. S here, and something I've mentioned actually earlier quickly, but let's highlight it here. It stands for, let me look my notes, Svedbergs. Svedbergs, okay. And or sedimentation rate. And this refers to how quickly it moves in the centrifuge. So the larger this number is, it means the larger the macromolecule. So the larger the molecule, okay? So in prokaryotes, we have 13S and 15S ribosomal subunits. And in prokaryotes, 13S and 50S ribosomal subunits. And eukaryotes, we have 40S and 60S ribosomal subunit. With that, I'm going to conclude. I know this has been a dense lecture and review for many concepts, not for you, but as well for me. And so I had to refresh with many of the readings, but I guess let's approach that as a way to refresh and make sure we have the proper foundational background to dive more into bioinformatics as we build up the pace of this course and as we go more in the future towards putting our eyes on data. We certainly need to appreciate the context of some of the very important concepts in molecular biology. Keep an eye on the assignments, discussions, and the, that are going to come next. And the quiz deadline is always Wednesdays, midnight. That is the deadline. Don't miss the midline. Come to the office hour. Okay, make sure you join the office hour. I want to meet you in the office hour and then discuss a few things. Know your background. How much of where should I focus in terms of tailoring my lectures, especially the coming lectures. So thank you so much and see you next week.